Welcome learners. Today we are going to deal the first topic, enzymes involved in DNA replication. So a fundamental property of all the living organisms is their ability to reproduce. Organisms produce their offsprings that are very similar to themselves. And this process of replication or this process of reproduction occurs in way by which a single parental cell divides to produce two identical daughter nucleus. And in this process, the genetic material must be duplicated or doubled prior to the cell division so that the daughter cell receives a full complement of the DNA genetic material. Replication. Replication means the synthesis of daughter nuclei molecules that are identical to the parental nucleic acids. Thus, the accurate and complete replication of the DNA is essential to the ability of the organism to reproduce. It is the basis of the biological inheritance. DNA is made up of two double strands, which are bound together by complementary base pairing. And during replication, these strands will get separated. And many enzymes participate in the process of replication. So they are DNA helicases, DNA polymerases, DNA clamp format formers, single strand binding proteins, topoisomerase or DNA gyrase, DNA ligase, and DNA primase. Helicases. Helicases are discovered in 1976 in E. coli, and these are vital for every living organism. And these are also known as helix destabilizing enzymes. As the name indicates, that helicase, it will unwind, it will separate the two strands of the DNA double helix. And these are the motor proteins that move directionally in the along the nucleic acid phosphodiester backbone, separating the two uh, annealed nucleic acid strands, thereby separating the two strands. And they will use the energy derived from the ATP hydrolysis. As shown in the figure, these enzymes helicase, uh, they are involved in the unwinding of the DNA. Since the DNA is a zip-like double strand, and they will uh, participate in the unzipping of the DNA. And next, DNA polymerases. So DNA polymerases are the enzymes that synthesize DNA molecules from the DNDPs, which are the building blocks of the DNA. And these enzymes are essential for the DNA replication because they work in pairs. Means there are two strands of DNA, as we see from this figure. There are two strands of DNA, and on each strand, one DNA polymerase can bind, and they will bring about the replication. And during this process of polymerization, they will read the existing DNA strand and adds the new nucleotides to it. And they also performs the proofreading and error correction activity also. So here two polymerases have bind, and they are participating in the process of replication. So this is the common reaction that occurs in the polymerization, and DNA polymerase are of major different types. In prokaryotes, we have three different types of DNA polymerases: polymerase 1, polymerase 2, and polymerase 3. And in eukaryotes, we have five types of polymerases. So polymerase 1 from the prokaryotes, it helps in the removal of the RNA primer, DNA repair, removal of the RNA primers. It also has the exonuclease activity from 5' and 3' N. Polymerase 2, it functions only in DNA repair, and it has 3' to 5' exonuclease active. Polymerase 3, it is the main enzyme of the replication. It performs the replication at a pace of adding 1500 base pairs per second, and it also possesses the proofreading and its editing capacities. And it has the 3' to 5' exonuclease activity. In coming to eukaryotic DNA polymerases, polymerase alpha, polymerase beta, polymerase gamma, polymerase delta, and polymerase epsilon are the five different types of polymerase in eukaryotes, of which the DNA delta is the major enzyme that is involved in the replication. And DNA clamp. These are actually formed by the beta subunits of the beta DNA polymerase, and these are also called as sliding clamps because they will help in stabilizing the protein. They will stabilizing the bonds that are formed between the template strand and the newly synthesized DNA. See here, we can see that this is a sliding clamp formed by beta subunit of the polymerase enzyme, and they will help in stabilizing the bonds. They will, until the new there is a complementary base pairing occurs, they will capture the old and new strands of DNA together. and single strand binding proteins. As the helicase enzyme unwinds the DNA, and these single strand binding proteins, they will bind to single strands of DNA to prevent the DNA leak. And these are about the almost double the number of helicase enzymes, and they consist of four subunits. Uh, these are the single strand stabilizing proteins. So this DNA cannot rewind again, and topoisomerase. So each cell has the uh, specific enzymes called topoisomerases. These are also called as DNA gyrases. They will stabilize the topology of the DNA, because as the helicase molecule unwinds, there is a torsion created in the backbone of the DNA, and this torsion is relieved by the enzymes called as topoisomerases. These topoisomerases are of two types: topoisomerase 1 and topoisomerase 2. Topoisomerase 1 transiently acts by breaking one strand of DNA at a time and rotating and releasing the torsion and rejoining the broken ends. Topoisomerase 2, they can break both strands of DNA at a time and rejoin together. And then DNA ligase. So DNA's replication on two strands occurs in two different forms. On one it is continuous, on the other it is discontinuous. The discontinuous replication results in the Okazaki fragments, and these Okazaki fragments are bound together or linked together by the enzyme DNA ligase. And the first enzyme is primase enzyme. This DNA primase is involved in the DNA replication. This DNA primase synthesizes the RNA primer of length 15 to 20 base pairs. And since DNA cannot synthesize, DNA replication cannot start on its own, it requires a RNA primer. That's why the DNA polymerase 2, where we have dealt in the earlier slides, that has the RNA primer, deleting the RNA primer. So this activity, this creation RNA primer synthesis is done by DNA primase enzyme, whereas the RNA primer is deleted by DNA polymerase 2. Primase is key important because DNA replication, because of no replication, DNA no polymerase cannot initiate the DNA synthesis on its own. It requires the DNA this one. So these are the enzymes that are involved in the DNA replication. Okay, thank you.